climate change, what climate change does to microbes and what microbes can do for climate change. So here you have the floor.
Thank you, Anna. Thanks everybody for coming. I've been at the other side of the TV double for decades at the Institute of Marine Sciences and never came to visit you. I finally went to Madrid and now I can come and visit you. It's a strange situation, but at any rate. First, I will begin with a commercial. We'll be back in 20 seconds. The commercial is for my new book of outreach about the ocean. It's just been published, and we'll have the official presentation on Friday at the Marine Sciences Institute at 12:15. And I don't pretend to become rich, but I would like the publishing house to sell enough copies that they will ask me to write another book. So buy it, give it to friends and to family and everything like that. And that's the commercials, and now the disclaimer, and this is a bit longer. Because of course, for some people, climate change doesn't exist, it's been terminated, and some of these people are pretty powerful, so it's difficult to really work against these people. And then the thing is that I don't know much about climate change, actually very little, and I'm a bit afraid of talking to you. I know that many of you work on these kind of things, and I don't, so I have to tell you why I'm giving this talk. And the reason is that in the last July, I was invited at the International Union of Microbiological Societies meeting, and they needed somebody to give a talk on bacteria and climate change. And they insisted on many different people, and everybody said no. And finally, the only silly guy they found that was willing to talk about this was me, and that was the reason to invite me. My reason to say yes was that we were in this wonderful hotel, and the important thing about this hotel is this thing here. All of this is where the infinity pool is. It's a swimming pool up there. And you can swim there. And the important thing is that this is the financial district of Singapore. So all these powerful guys who can do things to change climate change are working there. And for a few minutes, swimming here, you have the feeling that you have some power over them. Obviously, it is not true, but that was the reason I accepted to give this talk. But since I'm not expert about this, here is a book you can download from internet. It's in the American Society for Microbiology webpage, and you can download it, and it's very simple, but it has everything that needs to be mentioned about microbes and climate change. So the rest of the talk, I can just do things philosophical or something like that, and not into details. But I'll begin by doing a brief review of the methods that usually are used to study climate change. And of course, you know that there are all these different things that are happening with climate change, and we want to study them. And usually what we do is to use three different approaches to study climate change. One is having long-time series, right? We need to know how things are changing. Another one is mechanistic experiments to try to understand how things work. And then we have modeling. And I have to tell you that I will never give this talk to a lay audience. I will never give it to journalists. I'm giving it to you because I know you are working on climate change, and therefore you already know that it's for true. Okay, so let's begin with time series. These time series never seem to be long enough, and also they can be interpreted in many different ways. For example, here you have years since the 1980s, 2010, and this is the sea ice extent in the Arctic. And of course, you have a series of points, and you can fit three different lines and say, well, it was more or less stable, and then around the turn of the century, the ice began to decrease. But you can fit a different type of line, like this one, and then the points are the same ones, but now you have a straight line. So now the rate is not as fast as it was here, but it's more continuous. And of course, the interpretations of these are completely different. And even worse, you can fit something like this. And then the situation is even worse, because now the rate of decrease is increasing. So obviously, interpreting time series is quite difficult, and they never seem to be long enough. For example, this was published in Nature a couple of years ago. This is the stack normalized temperature anomaly in Antarctica, in a part of Antarctica. And as you can see, from 1980 to 2000, there seems to be a significant increase in temperature. And this is something that is understandable, since there is a temperature of the planet is increasing, this is happening. But then, when they studied the following years, now you find a significant trend that is cooling. So of course, interpreting time series is very complicated, and they never seem to be long enough. So the first leg has a crack, right? The second one is the mechanistic experiments, and this one is also fairly complicated. What you have here is experiments with sponges. This is one species of sponge, and they have been incubated at 3 different temperatures. It's only a 2.5 degree difference in temperature. And this is the way this sponge looks like. And you can see it's healthy here, but here it's in pretty bad shape. So just a change of a couple of degrees is enough to be really bad for this species. And the 3 points are different pHs. So this is current pH, and this is a bit more acidic, and this is a bit more acidic. Just maybe, I think it's 0.6 difference in pH. And you can see that at this temperature, nothing happens. But here, there is an increase in damage when the pH is very low. But here, at the highest temperature, increasing the pH actually ameliorates the situation. So this is something completely unexpected. And every different species reacts in a different way. There are about 14,000 species of sponges in the world. So does this mean we have to do 14,000 experiments to see how different organisms are going to react? Of course, if we want to model how things work in a precise way, we need to have this information. But it's incredible. It's 14,000 species, and each one may have a different way of responding to temperature and pH. This is another example of the funny things that a decrease in pH can do to organisms. This is the clownfish. This is the size of a larvae compared to an Australian coin. And they are challenged in these experiments with flow in 2 different flow chambers. The little fish is put here, and researchers record whether it goes this way or this way. So here you can challenge the fish with different substances. And what they did was to challenge the fish with different teas, different infusions. In the first case, it's just seawater, but seawater at current pH and seawater at 7.8 of pH. And of course, if it's only seawater, they go to each one 50% of the time, because you have seawater here and here. So they don't choose any one over the other, which makes sense. And if you lower the pH, nothing happens. It's the same thing. However, if you challenge them with this infusion of xanthodermon leaves, what happens is that normal pH, they all go for it. But if you decrease the pH, they don't go as much for it. The reason for this seems to be that this tree is a tropical forest tree, which is, grows near the areas where the water actually rinses it and brings these compounds to the sea in the areas where the animals are living. So the fish finds, it's a cue that the fish finds to try to find the symbiotic, the organism, the symbiont. If you do a tea of melaleuca, if it's normal pH, they never go for it. But if you decrease the pH, they go for it all the time. So this is confusing them. They're going for a cue that is not a good one. With grass, nothing much happens. And if you put the anemone, the pH doesn't matter. They go for the anemone all the time. So again, this is a very complex response. And the conclusion is that if pH goes down, probably little Nemo will not find his home, because won't be able to find the right cues. This is the third example of different experiments for climate change. In this case, what this shows is different genes that are overexpressed at lower pH. In the red is the acidified experiment, and the yellow one is the control. As you can see, these genes are overexpressed at low pH. This is logarithmic, so the difference is quite significant between the two. And if you look at what kind of genes these are, this is a scheme of a cell, and they're usually involved, for example, in electron transport chain. They're involved in photosynthesis. They're involved in transporters that use protons for transporting different nutrients, in proteorhodopsin. All of these proteins are involved in the proton balance of the cell. They either pump protons out or in. So it makes sense that if you do change the pH, these genes must be overexpressed because they probably have to do something with the protons. And if you look at different bacteria, this would be the same experiment, but looking at SAR11, Alteromonas, Flavobacterium, and Rhodobacterium. These are 4 very abundant bacteria. You can see that some of the genes that are overexpressed are the same, but some others are different. So each bacterium has a different strategy. Again, if we want to understand everything, we will have to look at all the bacteria that are there and see how they respond. But this is not the worst part. The worst part is the next one. Here you have this low chlorophyll. So this means that this was an experiment carried out with a marine sample where the chlorophyll was low. Now, if you do the experiment with high chlorophyll, then the only genes that are overexpressed are these ones, just a few. So the response is completely different. Again, the response of organisms to changes in pH and in temperature is incredibly complex and is different in different organisms. So the task that we have in front of us, we want to understand these things and be able to make good predictions, is fantastic. So again, we have another crack in another of the approaches. And finally, we have the modeling. And of course, we all know that models are what they are. And you have to have a pessimistic or optimistic model and a pessimistic model. And this range is huge. So models cannot be better than the data we feed them. So again, the third leg is also a crack. As I was telling you, I would never tell this to a journalist or to a layperson. But I think you are aware of the complexities of trying to make deductions about climate change. It's extremely complicated, and we have a lot of work to do. So that was the review of the methods. And now I'll ask 2 questions. The first one is, what can climate change do to bacteria? And the second one will be the other way around. What can bacteria do to climate change? And of course, I don't know. So I'm just making this up. And I hope that you, who are experts in climate change, at the end maybe can make suggestions or criticisms and this kind of thing. So in fact, this question will be more specific. So I will ask myself whether extinction of bacterial species is possible. And I will begin by telling you that there are a lot of bacteria. I realize you know that, but it's hard to imagine the dimensions of this number. If you take a cubic centimeter of seawater and you count how many prokaryotic cells you have, bacteria and archaea, you have 10 to the 5th cells. And if you look under the microscope, this is what you see. There's some sausages, croissants, meatballs, but nothing more. So this is what you see. Now, if you go to a 1 cubic decimeter, now you have 10 to the 8 prokaryotic cells. And this is the volume or the number of cells that usually people use to create gene libraries. This is a very standard thing of doing. If you consider this number of individuals, which is enormous, you have to think that maybe you are mixing ecosystems. If you were a plant ecologist and you were mixing the Valdivian rainforest with a very high diversity with the Patagonian forest, which has only 1 species, you mix them and then you analyze them, you get a completely wrong impression of the ecosystems you're studying. In microbial ecology, we have to deal with this. I mean, we know we take in so many individuals that we don't know exactly what's going on, and this can be a problem. Now, if we go to 1 cubic meter, now we have 10 to the 11th prokaryotic cells. And this is, for example, the volume that Craig Venter used for his pilot study of the Sargasso Sea. I'm sure you know that Craig Venter has been one of the most influential scientists at the turn of the century. He was the one pushing for the sequencing of the human genome. He did that with a private company, and he said he was going to do it in a few years, and people didn't believe him. But then he was getting very close, so then the public institutions had to do a consortium to compete with him, because imagine if the human genome were patented by a company. That would be really horrible. Okay, but at any rate, he was the force behind the sequencing of the human genome, and when he was tired of sequencing organisms, one week, for example, he had the cover of Nature with 1 genome and the cover of Science with another genome, just the same week. I mean, it was just amazing. So he got tired of it. He liked to sail. He had a yacht, so he went around the world sequencing the oceans. And in order to, in his talks, he had this picture of himself with the boat and his dog, and the dog had been sequenced also, and he was being sequenced at the same time. So it was really fantastic. And before doing this trip around the world, he did a pilot study in the Sargasso Sea. And this is the volume of water he used and the number of prokaryotic cells he was sequencing. If you were a plant ecologist and you were dealing with a forest with this number of trees, you would be dealing with a forest the size of Mexico. So again, the dimensions are hard to imagine, and we have to be aware of this, that this is not trivial. But then, here's Winter. This is his autobiography, and you cannot read this, but it says, "A life decoded." Of course, it's a play with words, because the code, the genetic code, blah, 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 and so on. But then he says, "My genome, my life." Certainly, he's no modest. But with this pilot study, he found thousands of species and 1 million new genes. Just these huge numbers. Now we move to the whole ocean. And we have 10 to the 29th prokaryotic cells. And this number is so unimaginable that you have to write it with all the zeros to really realize how big it is. 10 to the 11th, around here, is the number of neurons in our brain. And it's also the number of stars in our galaxy. 11 in here. And we still have all this to go. So this is a huge number. But yes, microbes are very small. Okay, let's compare them with whales. I've chosen the biggest whale for mass, the blue whale, but for number, I've chosen the smallest one because it's more abundant. So I'm overestimating the biomass of whales. Length, about 30 meters, 0.3 microns or 3 microns, something like that. Mass, but huge difference. Individuals of whales, this is the 2 mink whales together, and the individuals of bacteria, this is the 29th. So if you do the math, it turns out that the biomass of whales is this much, and the biomass of bacteria is this other much. So if you compare, the bacteria are equivalent to 214 million blue whales. And that means that the biomass is 536 times that of blue whales. So they're small, but there are many. And of course, since metabolism is proportional to size, so the smallest organisms are faster, in terms of metabolism, this will be even, the difference will be even bigger. What I'm going to say, I would never tell this to a journalist or to a lay person, but I tell it to you. If whales were to disappear, the economy of the ocean would not change. Their contribution is non-significant. Of course, if the bacteria disappear, the thing will be completely different. I insist, I would never say this to a journalist. Because of course, the cost of extinction of whales is a moral issue. It's not a question of ecology. I mean, of course, it's also ecology, but the main problem is moral. Okay, if we look at the microbial, well, the food web in the oceans, we have here sizes from 0.02 micrometers to 2,000 to 20,000. This is a fish larvae, like Nemo, in this previous experiment. So whales would be like 2 floors above, something like that. And here they are heterotrophic organisms and photoautotrophic organisms. So we have cyanobacteria, flagellates, dinoflagellates, diatoms, the classical phytoplankton. And here we have, again, bacteria and heterotrophic flagellates, ciliates, copepods, and so on. Now, if we measure the photosynthesis of all these organisms, it turns out to be, the estimates are, that is 50% of the primary production of the planet. So that means that half of the oxygen that you and I are respiring comes from marine microorganisms, and the other half for plants on land. And if you look at all of them and look at the respiration, then it's more than 95% of the respiration in the ocean is due to these organisms. So if you're interested in fluxes of CO2 and oxygen and influences on climate, of course you have to look at the microbes. That's where the action is taking place. So we have these many cells, but we don't know to how many species do they belong. And this is very easy to see the difference if we compare the number of species of plants on land. This is the colors indicate how many different species of land plants are there. So here in the Andes in Colombia, there are a lot of different species. In the Sahara, there are very few species. In the Arctic, there are basically no species in the further up. But you know, of course, there are all these problems with synonyms and the problems with identification and everything, but we have a pretty good idea that maybe they have 300,000 species of plants, and we know how many there are in different places. Now, if you go to the oceans and look at the primary producers in the oceans, this is what you see. The only thing you see is a little bit of color, indicating how much chlorophyll is there. But we don't have the slightest idea of how many different species are contributing to this primary production. So this contrast between the ocean and land is really striking. And the reason, of course, is that microbes are hard to see, and they have these boring shapes of, as I was telling you, croissants and sausages and so on. If we look at animals or plants that are similar to our size, it's relatively easy to tell that, well, they are probably two seagulls, right? And probably they are two different species. I mean, just by looking, it's easy. But if you look at bacteria, there's no way to tell how many species you have in this picture. So you have to resort to molecular biology. And many people tell me that microbiologists exaggerate the number of species of microbes because the molecular ribosomal RNA is overestimating diversity. So I'm going to compare the ribosomal RNA with the biological species concept. And you will see that I'm not exaggerating. This is a study, a beautiful study that was done in the Bay of Naples, where they have a very long time series of the phytoplankton, and they were able to see that there were two species of Pseudonychia. This is one species, and this is the other one seen in the light microscope. And you can see a little bit of a difference there in the way they interact with each other. I mean, to discriminate this, you have to really be an expert in taxonomy, because it's not easy. But this was well known. So by light microscopy, we had two species. Then they said, okay, let's compare with molecular biology. And they used the large subunit of the ribosomal RNA, and they found 1, 2, 3, 4 in this case, and 1, 2, and 3 in this other case. And again, here's where a classical taxonomist would tell me, see, you are overestimating the diversity of the microorganisms. But then they said, okay, besides the ribosomal RNA gene, let's look at the interspacers. You recall that we have 3 ribosomal RNA genes, the small one, 5S, the 16S, well, the bacteria, 18S for us, and then 23S more or less. So we have 3. And in between those 3 genes, there are 2 interspacer regions. And these interspacer regions usually do not code for anything, so they can change a lot. The ribosomal RNA cannot change very much because its function is so essential. It's forming part of the ribosome, the machinery to make proteins, that it cannot change very much. And that's the reason why we can compare all living beings with this gene, because it hasn't changed very much. But the interspacer region changed a lot. So if you want to really be fine in the taxonomy, it's better to, just for one group, it's better to look at the interspacer. And as you can see, in this case, there were 1, 2, 3, 4, and 5. In this case, there were 3. So there was a little bit more than with the ribosomal RNA. Okay, the advantage of this example is that these people in Naples have a lot of clones of this algae that they have isolated through the years. And diatoms can have sex, so you can test the biological species concept. They crossed all the different clones to see what would be the biological species that they will find. And it was exactly like in the case of the interspacer region. So the ribosomal RNA was actually underestimating the diversity according to the biological species concept. Once they had this information, they went back to the clones and looked very carefully with electron microscopy, and then they saw tiny changes in the patterns of the frustules of the diatoms. So once they knew, they could actually see morphological changes in them, and then they could define all these different species. So I think it's a beautiful example of comparing molecular and classical taxonomy, and how, when you look at them carefully enough, they usually end up fitting together very well. But at any rate, my point is that by using ribosomal RNA, I'm not overestimating diversity. In fact, there was a paper appearing last year in which they found this relationship between the number of individuals and the number of species, in this case of operational taxonomic units, because it's defined with ribosomal RNA, not with other things. So these are all experimental data, and you say, if you extrapolate from this to this is crazy. But my colleague, Susana Manrubia, did all the calculations again with their own data, and it was good. It was statistically significant, so we have to believe it. And then if you go, for example, how many different species are there in the human gut? And this is not the gut of one of us, it's the guts of all the human beings. When you go to this and you find there's 10 to the 6 species. How many are there in the ocean? You go to the 10 to the 29 individuals, and you go here, and it's 10 to the 10th species, or to the 11th, more or less. And if you go to the whole Earth, it turns out there should be more than 10 to the 12th species of microorganisms. This is amazing. I mean, you are free not to believe it, because obviously it's an extrapolation, and it's a hypothesis, but certainly suggests that there can be a lot of different species. And so if you have this number of cells and this number of species, how many cells can there be for a species if all of them have the same number of individuals? Well, each one could have all this number of individuals. So there is plenty of room for all these species. It's not a problem of having few individuals. Okay, the problem is that to analyze all these species is that they are not found in equal abundances, of course. Like all other living beings, they have these rank abundance curves, where the most abundant species is here, and then the second one, and then you go back to the rare ones. And the problem is that the rare ones are so rare that only until recently have we been able to find them. First, we were doing fingerprinting techniques, and this would give around 30 taxa per sample. Then with the cloning, and then we could have maybe 500 taxa. And then with metagenomics and massive viral sequencing, we can go down to many 20,000 or 30,000 species. But we don't know if we still have to go further, because the techniques are where they are. But at any rate, it allows us to go much farther than before. And this is the shape, one of these rank abundance curves from Blanes, from the Sea of Blanes, where we have the most abundant bacterium is SAR11. This SAR11 is the most abundant bacterium in the oceans all over. And it has 11% of all the sequences in this sample. And then they go to the greatest one. For example, this one has 0.003%. So it's really difficult to get these organisms. But we can isolate it in pure culture. So that we have two ways of accessing this diversity. One is the massive sequencing, and the other one is the culturing. Unfortunately, we cannot culture many of them. We can only culture some of them. But at least it provides some of them. And then the question is, if this is the situation, that we have this kind of rank abundance curve, what do they do, the bacteria in the different places? Well, of course, you would think that the abundant ones are the ones significantly contributing to carbon flow in the ecosystem, right? And then the intermediate ones probably contribute to some element flow, for example, nitrogen fixation. This is an essential process in the ecosystem, but it's carried out by bacteria, which are not very abundant. So these probably are somewhere in here. And then the rare ones have a significant contribution to biodiversity. I don't know whether you recall that Margalef was making a distinction between diversity and biodiversity. Biodiversity was like the alphabet or the dictionary of a language, and biodiversity, and diversity was the words used in a particular book, right? So in this case, they contribute to biodiversity, but the contribution to the diversity of the ecosystem is minor, because they're very rare, and probably they do not contribute to carbon and energy flow. This is a beautiful example of how this functions. This was done when the Prestige sunk, and all of a sudden the government realized that they didn't have any experts on oil degradation, or not enough. And all of a sudden there was a lot of money to the research on oil degradation. Of course, all of us became immediately experts in this kind of field, so to get some money and do something. And this was done by some of my colleagues in Vigo and in Barcelona. And what they did was to put these mesocosms with water from the Ria de Vigo, and added different amounts of these oil derivatives from the Prestige oil. And then they followed what happened with time. And this is an example of what they found. This is days from the beginning to day 8. This yellow curve is the amount of oil derivatives. So at the beginning they added this much, and it disappeared. The black is the abundance of this one bacterium, Psychoclasticus, the abundance in the control without oil, so it's indistinguishable from 0. And the blue is the concentration on the experimental mesocosms where the oil was. So you can see that the oil decreases exactly as the Psychoclasticus increases, and then once the substrate is finished, it goes back down, right? So it came out of this rare biosphere, this rare part of the curve, became very important in carbon flow in the ecosystem, and then disappeared again in this rare part of the biosphere. And the interesting thing is that if you increase the amount of oil derivatives, you increase the amount of bacteria growing. So this is a very nice experiment. Everything fits together very nicely. So this is probably happening in nature constantly, with bacteria which are in that part not contributing to anything to carbon or energy flow, but being there just in case something happens. And this is what gives microbial communities the capacity to adapt to many different situations. The other interesting thing is that once this population declines and goes into this rare part, it has several advantages. One of them is that it crosses a threshold below which viruses do not find it. As you recall, viruses move by Brownian motion, and they have to, by chance, find a susceptible cell to infect it and reproduce. The thing is that if the concentration of susceptible cells is lower than this number, the viruses will not find the cell. So you are, by being rare, you are protected from viral attack, which is extremely nice. Of course, you're not doing much, but at least nobody is getting you. And the second threshold is one of predation. And this is not as clear, because predators, in principle, will also eat bacteria that are rare in proportion to their abundance. But the thing is that predators are not silly, they're not stupid, and so they prefer big cells, because the big cells are more nutritious, they're usually actively growing, and they have more nutrients. So usually they select the biggest cells. So by being rare, you are protected from the two main mechanisms of death for bacteria. So in principle, you could stay there for a very long time. And now we're getting back to the question of whether bacterial species can be extinguished or not. One example of how rare you can be and still be there was done by my colleague Pierre Galland in France. He found this bacterium, Archobacter, as one cell in 10 liters of seawater, and was still able to grow and contribute significantly to ecosystem processes when things changed. And he drew this cartoon where you have one bacterium and one human being, and if you have one in one drop of seawater, it's rare. It's like having a person in a soccer stadium. And if you have one in 10 liters, so what he found is like having one person in the whole world. So this is very rare. And yet there are thousands of species that are being rare with many genes that potentially will be useful for whatever happens in the environment. Of course, you will say, okay, this bacteria will be there, but what about bacteria living with animals? What about the symbionts? If you have a problem with corals, for example, this is the healthy one. These are these, what's the name in English? It's the white ones? Bleaching, exactly. These are bleached corals. So they have expelled their symbiotic dinoflagellates. If these corals become extinguished, those microbes that are specific for them will supposedly also become extinguished, right? And that's really a possibility. What happens usually is this is the cell of the coral with the nucleus. Here's the dinoflagellate with its chloroplast and mitochondrion, and here's the mitochondrion of the coral. What happens is that when temperature increases, photosynthesis also increases, just because of biological processes. And then a lot of oxygen is generated that the coral cannot deal with. So this generates all this ROS and oxidative stress and calcium imbalance and everything. So that by lowering the pH, there's a good chance of extinguishing coral species, and then maybe the symbiotic microbes will also become extinguished. And these events have become more increasingly frequent, so this is something likely that may happen. All animals have a microbiota. And for example, here you have a sponge, a coral, and a fish. And these are different bacteria. In blue are the bacteria present in the water surrounding the animal. In black, the bacteria in the animal. And as you can see, they're different, right? Because this is a very specific niche, and so it requires a specific bacteria. So yes, if this sponge becomes extinguished, maybe this bacteria won't be able to survive. However, it must be considered that things are really interesting. This is a case of a squid, the bobtail squid. It lives in tropical waters, and it usually spends the day in the sediment, just hiding. And at night, it comes into the plankton to feed. And of course, if you're in the tropics and there's moonlight, or maybe even the stars, and a fish, a predator is looking under you, it will see your shadow against the light sky. So what they do is to have light organs with bacteria that produce light to compensate this light coming from outside. And therefore, they're safe from, well, relatively safe from predation. So what they do is with these bacteria is that they feed them actually. So they can grow and then they start to emit light. And they can regulate this, how much light they emit. But if they didn't do anything, these bacteria would keep increasing and their light organs will become huge, and this is not possible. So what they do is that when the sun comes up and they are going to hide back in the sediments, they expel most of these bacteria into the water. And then during the night, they just grow them again for the next night. And as a result, you find this bacterium, which is Vibrio fischeri, you find it in the water. And this concentration, 10 to the 3 cells in a milliliter, is being part of that rare biosphere. So the bacteria that live as symbionts on animals are also found in the water column, but in extremely low abundances. And there is a reason for this. The reason is that the newly hatched squid, the baby squid, do not have this light organ formed yet, and they don't have the bacteria. So they have to start filtering water, and when they find one cell of Vibrio fischeri, they then begin to develop the light organ and to grow this bacterium. And the recognition is very specific. It can only be this strain. And if you put a different strain of Vibrio, this will not happen. So they need to have this bacteria in the water. They need it for the next generation. And therefore, this bacteria will be there for a very long period of time. Of course, researchers are known for being cruel with animals. And what the experiments they've done is to provide the squid with Vibrio fischeri, so the surface of the organism is the same, but mutated in the genes that produce light. So it's a Vibrio fischeri that does not produce light. Of course, the squid detects it's the right bacterium, so it incorporates it, develops the light organ, and what do you think happens then? You've guessed already? What happens is that the squid is not silly and expels the bacteria that are not emitting light and looks for another one that will emit light. So they are really smart. Then some people have looked at the concentration of this Vibrio fischeri in the water. These are different places, but let's look at this one. This Botany Bay is a bay where the squid was very abundant. And this is the abundance of Vibrio fischeri in the water in the morning when the squid throws away the bacteria, and this is at night when it doesn't. And this is another day. So this abundance keeps changing, keeps fluctuating during the day and the night. But the message is that the bacteria, even those that are associated with animals, are there as part of the red biosphere. The other thing is that bacteria can disperse to very long places. And this is an example of the Saharan desert and the Canary Islands. And you can see dust storm. Then my colleague, Emilio Casamayor in Blanes, collected sediment from here. And then he went to the Pyrenees to, I think this is Steinredo, that many of you are probably familiar with. And he then collected also the precipitation. He had this kind of instrument. Here is collecting dry deposition, and when it rains, there's a sensor that this moves and collects wet deposition in a separate place. And then he looked at the bacteria. And the interesting thing is that he found some bacteria which are practically the same from Mauritania and from the Pyrenees. From Mauritania and from the Pyrenees. So these bacteria who are inhabiting the soils of the desert are able to travel to a different continent and be deposited on this lake. And then one doesn't know what they do there. They obviously become part of the rare biosphere of this lake. Emilio did other experiments that were very nice. If he incubated water with this bacteria could not compete with the local bacteria, so they didn't grow. And this became rarer and rarer. But if he incubated them with the water from the lake, where the bacteria had been filtered out, so it was the water but not the bacteria, then they could grow. So this is a very nice case of being invasive or not. So this also happens with bacteria, but at any rate they can travel very far away. And then this is my favorite example. We have the ice caps, and the ice caps, well, Antarctica and Greenland, they are collecting a sample of the atmosphere every year, right? There's snow coming on dry deposition, and this is preserved on the ice. It's frozen, like in a freezer, of course. And then slowly this ice accumulates, and this layer of the present just keeps going down, and eventually moves away from the ice cap, and the ice is melted, and these bacteria are released in the ocean. Could it be that they are still alive or not? Well, people have looked at these places. They have taken cores, and this is one example. This is ice from 500 years ago. This is ice from 157,000 years ago. And these are microorganisms that were able to grow. So they were alive. They're frozen, but they were alive. And this has been done even with, I believe, almost 1 million years. So if you can be preserved for 1 million years, I mean, this is a time machine, right? It's bringing organisms from the past to the present. If you can stay there for 1 million years, you can go by a couple of glaciation events with no problem. You can survive a lot of things if you can stay there for 1 million years. So I think that disappearance of bacterial species is possible, but very unlikely. And maybe that's why there are so many different species, because speciation is quick, but extinction is not very unlikely. Oops. Okay, and then the next question is, what can bacteria do to climate change? And of course, you know about the Gaia theory. The idea is that the living beings are changing the planet to make it better for life. This has been very criticized, but I like it because it has this Greek goddess, and we'll see that there is a counterpart and everything. And I'm just using it as a metaphor of mechanisms that the biota uses to try to preserve the conditions. One of them is based on these marine microbes. Algae have DMSP, dimethyl sulfonopropionate, as an agent to maintain the osmotic pressure. And when they are eaten or when they die, this is released into the water, and the microbes convert it to DMS, dimethyl sulfur. This is volatile and goes into the atmosphere, is oxidized into sulfate and sulfonate aerosol, and these aerosols act as cloud condensation nuclei. So if there are a lot of algae, there will be more clouds. If there are more clouds, less solar radiation will reach the surface. And then that means there will be less photosynthesis and less organisms. So here you can see a feedback mechanism whereby some of the temperature changes can be sort of balanced. Again, this has been criticized, but many of these aspects, I think, can be trusted. Another example is the biological pump. The algae fix CO2, and it's respired by many heterotrophic organisms, but a portion of the CO2 is buried in the sediment and disappears from the atmosphere. This is another way of decreasing the concentration of CO2 in the atmosphere. And they can also affect not just the biology, but also the geology of the planet. For example, these algae have these calcium carbonate plates. When they die, they sediment to the bottom, they transport it and convert it into rock, and this emerges many millions of years afterwards as the rocks, the white cliffs of Dover. So the microbes can also affect the geology of the planet, which is, when you think of it, it's really amazing. But of course, we have the counterpart of Gaia. We have the Medea theory. In this case is that no, the microbiota is not willing to share the planet with us and tries to get rid of us periodically. This was presented by Peter Ward, and he claims that there have been several episodes in which the complex life almost disappeared from the planet. For example, this methane poisoning, 3.5, well, there were not big organisms at that time, but maybe life could have been disappeared. Or this, the Great Oxygenation Event, this is one we know well. There were snowball Earth occasions in which also big organisms could have disappeared. And he claims that also some mass extinctions recently produced by hydrogen sulfide. So his claim is that the biota is not acting to make life better, the planet better for life, but to make it worse for life, or at least for part of life. Maybe the current climate change is a way for the microbiota to get rid of us. And that would make sense with this Medea theory. But if we look at this one, this is time, the origin of life and present, and oxygen all of a sudden begins to increase. And this is because cyanobacteria invented oxygen in photosynthesis. And that changed the Earth. I mean, it changed the atmosphere of methane and CO2 to the atmosphere we enjoy today of oxygen and nitrogen. This is no doubt the biggest change, the biggest global change that the planet has experienced. And it had tremendous consequences for all the living beings. So I asked myself, we are now in front of another change. If I had been a microbial ecologist in the Hadean period, before this happened, what would have I been able to predict about it, right? And of course, one thing I could predict is oxygen will increase due to the sedimentation of organic matter. This is the biological pump. Since not all the CO2 goes back to the atmosphere, the oxygen will remain and will increase. Okay. Oxygen is such a powerful poison that most life forms will disappear. This is another prediction that probably happened. I don't know, but it's a prediction that I think I would make. And since cyanobacteria have learned to handle oxygen, probably other bacteria will also learn. So I think some predictions about climate change, global change, are possible. However, I could have never predicted this. The fact that some organisms that were not able to deal with oxygen would actually eat one of the few bacteria that learn how to deal with oxygen, actually to use it for obtaining energy and convert it into mitochondria, and actually eat one of those photosynthesizers and also incorporate it to make current eukaryotic organisms. This would have been completely impossible to predict. So some things cannot be predicted. And I like very much the sentence from the movie Jurassic Park. You remember the mathematician, the guy dressed in black, really cool and so on. And he has this sentence saying that life finds a way. So I'm sure that even if the change is very dramatic and even if we disappear, which is very unlikely, but even if that happens, of course, life will find a way and microorganisms will be doing all the things. So, which leads me to the final question. If we were able to understand how microbial communities work, if we really understood how organisms interact with each other to assemble these communities, microbial communities, maybe we will be able to do something like terraforming and convert it from a game, into something useful. Maybe we could go to Mars and change it to make it livable. And maybe that would alleviate some of the pressure they're putting on planet Earth. And this, I think it's a worthwhile objective, even if science fiction today, even if it's extremely dangerous, because of course, you would have to do a lot of tests before actually doing it. I think it's a worth purpose to do. So the conclusion, which I would tell the journalists and the general public, and also you, is that is this metaphor. We are driving the Earth as a racing car at full speed, while still reading the instructions manual. So I think it would make a lot of sense to take the foot off the accelerator. Thanks for your attention.
Yes, Carlos, yeah. There are plenty of questions. Thank you very much for your very interesting talk, very inspiring. So, for example, now you are talking about predictions, your last message. Why you wouldn't consider a abrupt shift? Because all your predictions seems to be very progressive and linear. So, and the change to eukaryotic cells became a really abrupt shift. Mm-hmm. Would you imagine now an abrupt shift in front of this climate change era?
Well, of course, I was a bit, I wasn't fair enough because that took place through millions of years. And now we're confronting a change in tens of years, right? So this would not happen in this the same time period. But so the question is whether I could make predictions, not changing linearly, but...
well, the thing is that we should change the paradigm. We should change the way we think about changes. So maybe we should be more brave and consider this kind of abrupt shift. So not so linear and easy and progressive. It's one thing, but coming to more real things, mm-hmm. or more things that we are living now, I wonder, we always discuss which is the role of the oceans in the uptake of CO2. So you seem to favor the message of CO2 uptaken by cyanobacteria, by organisms. Mm-hmm. But there are other people that discuss continuously about the physical-chemical uptake of CO2. Could you explain us which is the status of that?
You mean the chemical pump?
The chemical pump.
Mm-hmm.
So
well,
could you summarize for us, because we are terrestrial guys?
Actually, it's hard for me because this is biogeochemistry and I'm a microbiologist, so it's also hard for me. But the idea is in the case of the chemical part, of course, this is very complex equilibrium between CO2, bicarbonate, carbonate, and calcium carbonate and everything. So if you're able to transfer the CO2 to calcium carbonate, then you're able to precipitate it purely chemically, by chemical equilibrium, then you have a chemical pump. And if it's buried in the sediments, then it's okay, it's there for some time. And in the biological case, it's just because not all the primary production is transformed by the heterotrophs, the fish and the copepods and so on, back into CO2. But part of it sinks to the bottom and is also buried. So then it's, again, carbon that is going to be there for quite a long time.
Yeah, but the point is, which is the relative importance?
I don't know. The problem is that, I mean, people do a study in the Central Pacific, and one time of the year, and you have one number. But then you go to the Arctic in another time of the year, and then you have a different number. The problem is that it's like the experiments I was showing. We don't have enough measurements with enough periodicity in enough places to really make a global balance. So people, of course, jump into making a generalization, extrapolating from too few data. And that's not surprising that people come with different numbers. But there are many people, not many, but many people who are, not many, some people who are really into making these general extrapolations because, I mean, they get published in Nature, so it's really nice, you know, making it and so on.
We see people claiming that it's just a matter of the carbon dioxide bicarbonate system, and other people that claims that it's everything biology. So we don't know what to think.
Somewhere in the middle, for sure.
Okay.
So, very nice talk, really. Many thanks for the very stimulating ideas. So when you think about these large scales, you know, in these big numbers and the small numbers, and then you think about even space, you know, other planets. So how do we conceive? How do we think of life when, in terms of the laws that really limit these processes at such different scales? Is it the stoichiometry of life, or is it some kind of scaling of metabolism that?
You mean in other planets, or?
Well, I take that as an example, but we made also example of comparison between whales and bacteria, you know, and so they're so different, and yet you know they live in the same planet, you know, and they do live in the same place, you know. So I don't know. Can you think in those very broad, almost biophysical terms, and tell us something about this sort of regulation?
I'm afraid I don't understand the question very well.
Well, I'm thinking, is it you know, if you think about a different planet, is it the stoichiometry of life that limits the scaling of the number of organisms, the size that they reach? What is the fundamental limiting concept or law that controls life? You know, can we think in those terms?
I don't know. However, one thing that I find truly interesting is that life finds new niches all the time. There's one example, one experiment that I like very much that was done with a continuous culture of Escherichia coli. So it was fed with glucose. So this is the most boring ecosystem on Earth. It's just glucose and one species of bacterium. And it was grown by several thousand generations. And at the end, there were 3 different populations. One was taking the glucose and excreting, I think it was glycerol maybe, and acetate, the 2 different compounds. Another one was specialized in the glycerol, another one in the acetate. So you had the appearance of 3 niches where there was only one. And that's because life is constantly, you know, exploring these possibilities. And I assume that that's why we have whales, because life has explored so many different possibilities that eventually fits more things where they look like there were no more space for other living beings. But I realize it's not a very good response to your question.
I have 2 questions. One is related in some way with the original question. That is, you are a microbiologist, we are ecologists, we deal with concepts, you deal with very small organisms. And microbiology has changed very fast from the incorporation of these molecular techniques. And we ecologists are very aware about that. And my question is, which concept of ecology that is a discipline with a lot of theory do you find as a microbiologist that are incorporating in your normal working and to frame to understand your discipline?
Well,
so my question is just to find bridges between the microbiology and the ecology.
This is very frustrating because we've always learned the ecology of the large organisms, the animals and plants. And I mean, I'm an ecologist, but microbial, so I admire these concepts of ecology. And I've always tried to see whether I could apply them to microorganisms. And there's always something that prevents you from actually interpreting the things in the same way. But having said that, there are many particular examples when things work very nicely. I mean, the famous experiments of predator and prey were done with microorganisms, and that has become one of the common laws of general ecology. So when you have to deal with experiments that can be done with a couple of species or 3 species maybe, and which need a long time, then microbes are a good model. But otherwise, interpreting, for example, communities, of course, is also complicated for plants and animals. But for microbes, we're just beginning to see what's there. I mean, 10 years ago, we could only find 100 species in one sample, and now we can find maybe 20,000 in one marine sample. So we're just describing what's there, and we're very far behind in terms of applying ecological theory. But it's something that we look forward to.
Yeah, that brings to the second question, but before going there, what you are describing is a kind of way to reductionism, in the sense to understand each of the pieces of this microbiology wall. And what we find is the number is huge, and I don't know, maybe eventually the computers will deal with that. But anyway, my philosophical question that I also said somewhere is if we should go back to some of these moralistic concepts to deal with this big complexity and big data. This is one question. I have the last one.
Modalistic, you said?
Modalistic, no thinking, no frame to reconvert and to take this biodiversity. Diversity is an holistic concept. So move back to this kind of concept just to deal with a huge, because what I feel is that we are going to describe each piece of the universe, but I'm not sure that will give us the tool to understand the universe.
Yeah, who knows? But I mean, in terms of ecosystem function, that was the thing that could be done very well in microbial communities. So you could measure primary production and respiration and everything very easily. So that in that respect, in microbial ecology, I think we followed the ideas of general ecology without problems. But in terms of, for example, in community assembly, we are struggling with it because we don't have still the knowledge to really test ideas and this kind of thing. And that happens with many of the general ecological theories. But as I say, I think most microbial ecologists would like to be able to interact with ecologists to try to apply these theories to microbial communities. And that's something that we would love to do.
And just to finish, my last question is, what do you think is the next frontier, the next edge of microbiology in the sense of where you are?
It's genomes and transcriptomes. It's fantastic. It's like, the genomics has been like opening a window into. No, let me put it this way. Before genomics, exploring life was like going in the dark through a forest with a spotlight, and so you could walk along a path, but you didn't know what was there 10 meters away, and you would see only the thing you could put your light on it. But now it's like the sun has come up, and now you see this map of life. The problem, of course, is that it's so huge that we know very little about it, but certainly has completely changed the way of looking at life, and it's changing the way that we're doing microbial ecology for sure. The number of genes that we find, I remember I'm getting too excited, but when I was studying biology, I remember, and actually maybe Ferran was there too, in La Salle. We had, all of a sudden we had one of the teachers that actually opened a mouse for us. We opened and you don't remember? For me this was incredible because all of us, we had the books of biology, and one chapter was the digestive tube, and you had this face sideways, and then the digestive tube, and then the anus. Then another chapter was the skeleton, another chapter was something else. And all of a sudden, we opened that mouse and everything was there together. You could see the intestine with this membrane and with the little veins and arteries and everything, so delicate and so well integrated. And this is, to me, is happening again with genomes. Now, we knew for many years that this organism has this gene and also has this other one, but now we have the whole genome. And you can see how the genes move around, or how they're together, to work together, how they are regulated, and this is again is like the same experience of discovering a new window into biology. So it's really a fascinating time to to be doing biology. For those of you who are young, you have a fantastic future.
Lots of
sorry.
And that we talk about genomics. Do you think it's comparable, the concept of species between prokaryotes and eukaryotes?
I don't think we know what the species is in the case of eukaryotes. There are at least 30 different definitions, and the most classical one, the biological species concept, has only been tested for a very minor percentage of all the species of animals and plants. And one example is the seagulls. The yellow-legged seagull here in the Iberian Peninsula interbreeds with another gull in the Eastern Mediterranean, and this one with another one. And the one in America does not interbreed with this one. So what is the species there? How do you break it up? And of course, I mean, this is very logical because evolution is a movie, and we're just looking at one picture, one frame, the frame of the present. And some species are very clearly different. Some others are in the process of differentiation. And I think this is the same for bacteria, for prokaryotes. Now, you're right that there is another difference. In the case of microorganisms, there is this lateral gene transfer, and this confuses things quite a bit. This is very easy to have new genes incorporated or lost. However, mostly the core genes, the ones for the DNA replication, for protein synthesis and so on, seem to have a vertical inheritance. And so in that case, you have the same situation that with animals and plants. You have a tree, and when the branches are just barely separating, it's difficult. When they have separated it more, it's easy. If you talk about how the species, how do they interact in ecology, then that's more complicated, and I don't know what to say in that case.
But you have this axis with number of species.
Yeah, well, OTUs.
Well, which apparently the ideas were clear in prokaryotes. So maybe were all the genes.
No, in
the question is, it's just genes, or because for eukaryotes things look much more complicated because you have more things to integrate.
You mean in the definition of the species?
Yeah.
No, in principle, if bacterial taxonomists are also very demanding on the information you have to provide. And you have to provide, for example, now you have to provide the whole genome of the bacterium. And also you have to provide a phenotypic test that would allow you to differentiate this species from the closest relative. So there are quite a few things that you have to do to properly describe a species. That's why there's so few described, because it's very, very difficult. But what I'm showing here is what you find in nature based on the ribosomal RNA. And the assumption until recently was that you would assume that anybody who has more than 90% similarity in their sequence is the same species. Now we've realized that's not the case. You have to go to 99%, because otherwise it's very, very different. No? But that's only a practical, as the name indicates, operational taxonomic unit. So I would not equate that with a species.
Just the last question. You gave a number for prokaryote biomass. Do you have a number for eukaryote biomass to compare?
No, you're right. That's a good exercise. I'll try to do that.
Because when we say individuals in eukaryotes has many cells, and even a cell has several prokaryotes together.
Yeah. When we have in a cubic centimeter, we have maybe a few thousand eukaryotic microbes. So, and they're much bigger. So I'll do the calculation. That's a good idea. Yeah.
So last question, I have two questions here. Giuseppe is waiting for, and then you.
Okay, I know. I am really excited also. But like Mark, I am a little bit skeptical about this use of OTUs as species. You know, this is a controversy. And I am afraid that we can overestimate the number of species depending on the way we define them. But you already discussed this with Mark. The other thing that I was excited to hear from you is that you think that bacteria are more unlikely to get extinguished than plants and animals. Why is that? Because it's something that we worry about a lot as biogeochemists. For example, these days we see continuous input of nitrogen in biosphere, and we wonder what's going to happen with nitrogen-fixing bacteria, for example. Is it going to change the community of bacteria dealing with the global nitrogen cycle, for example? And we wonder, these species are going to be extinguished, these ones, they are not. So now I was surprised for your statement, bacteria extinguish much more likely?
That was my idea, but I'm not sure about it.
Okay, but so can you go into it?
Well, the idea is, as I was explaining, is that there are many individuals that they can stay there, being rare for very long periods of time. They can be preserved in the ice caps. So it's really difficult to get rid of them. Now, of course, if you change conditions, the bacteria that will become abundant from that rare part will be different. But I'm sure you will have somebody fixing nitrogen. I would put my hand on fire. That,
yeah, well, they are still very flexible. They are more flexible than more complex organisms like you.
Yeah, and especially because there are so many different, now I won't say a species, but so many different whatever. Each one with thousands of genes, and they're in different combinations. So there's always one that will find a way to grow.
Yeah.
So I think it is the last one.
My question is if I is close to the last one, but I was curious about this. You're saying a lot of extinction. Extinction is possible, but less common in bacteria. So since you have been studying these extreme habitats, have you seen an extinction in these high salt habitats or very dry, very dry, extinction of bacterial species?
I don't think I could. I don't see what experiment could I do to see whether a species has been extinguished. The problem is, let's say that you go to this hot spring and you determine all the species that you can in that hot spring, and you find, I don't know, hot springs, maybe there are 300. Okay, then you go back several years later and you try to see whether one of those 300 has been extinguished. The question is, maybe you won't find some of them, but if instead of collecting a sample of this size, you were collecting a sample of this size, most likely you would find them. So I don't think it's a way of experimentally determining that one species has been extinguished. In the ocean, you would have to sequence the whole ocean, and of course that's not possible. So it's a speculation.
What about functional genes?
Sorry?
What about functional genes? Do they disappear from a place like the one you were describing?
What happens is that if a function is not needed anymore, like the Psychoplasticus with the oil, that particular bacterium will go back to the rare part and will stay there. Now, of course, if a very long time passes without another event happening, maybe it will be so rare that it will become extremely rare, but otherwise it will be there.
Okay, thank you very much. We see that the world can continue as long as the bacteria are there.
Yeah.
Okay.